Metagonimus yokogawai or the Yokogawa fluke is a species of trematode or fluke worm in the family of Heterophiidae. It is the smallest intestinal fluke. It was found in Asia and Siberia and is known for the causative agent for the disease metagonimiasis. Metagonimus yokogawai was originally described by Katsurada, reported on 1912. Snails are the most frequent intermediate host for Metagonimus yokogawai. It comes from the genus of Semisulcospira, which is a genus of freshwater snails. The distribution of Metagonimus yokogawai is geographically distributed in Korea, China, Taiwan, Japan, Siberia, Indonesia, Israel, and Spain. It was reported that the most common intestinal trematoid species reported in humans are in the Republic of Korea, since they are more likely to consume raw freshwater fish. The habitat of Metagonimus yokogawai spans freshwater environments encompassing freshwater snails as intermediate hosts, freshwater fish as second intermediate hosts, and the digestive tract of humans and other mammals as definitive hosts. Once infected fish are consumed by definitive hosts, which include humans and various fish-eating mammals. Metagonimus yokogawai completes its life cycle in the intestines of this host. So the parasite matures into adult worms, reproduces sexually and produces eggs, which are then passed out with the host feces. So they become adults and reside in the small intestine of the definitive host. The morphology of Metagonimus yokogawai is characterized by several distinct features. So in eggs are very small. Eggs have a smooth, hard shell that is transparent and yellow-brown in color and an ovoid egg shape. So they are about 26 to 28 micrometers and 15 to 17 micrometers width. So the egg also has a very slight opercular shoulder. The life cycle of Metagonimus yokogawai begins when an adult releases fully embryonated eggs, each with a fully developed residuum, and eggs are passed into the host's feces. So after ingestion by a suitable snail, which is the first intermediate host, the eggs hatch and release myricidia, which penetrate the snail's intestine. So the myricidia undergo several developmental stages in the snail. Many cercariae are produced from each radiae. The cercariae are now released from the snail. So it is now encysts as metacercariae in the tissues of fish, which is the second intermediate host. The definitive host becomes infected by ingesting undercooked or salted fish containing metacercariae. After ingestion, the metacercariae exist attached to the mucosa of the small intestine and mature into adults. Infection occurs when the metacercariae, the infective stage of Metagonimus yokogawai, are ingested along with raw or undercooked freshwater fish. Once ingested, the metacercariae exist in the duodenum, releasing juvenile flukes. These juvenile flukes migrate through the intestinal wall and enter the bile ducts and mature into adult flukes. The adult flukes then produce eggs which are passed in the feces, completing the life cycle. This is characterized by gastrointestinal disturbances such as abdominal pain, diarrhea, and nausea. In immunity, upon ingestion of metacercariae, the juvenile form of the parasite, the innate immune system is likely the first line of defense. Components such as macrophages and dendritic cells may recognize and phagocytose the parasite or its products. Activation of innate immune pathways triggers the release of pro-inflammatory cytokines and chemokines, recruiting other immune cells to the site of infection. Diagnosis and treatment of Metagonimus Yokogawai infection typically involve several steps. For the diagnosis, clinical evaluation, patients presenting with gastrointestinal symptoms or suspected parasitic infections are evaluated clinically. Symptoms may include abdominal pain, diarrhea, nausea, and vomiting. Next is laboratory testing or the stool examination. Microscopic examination of stool samples is the primary diagnostic method for Metagonimus Yokogawai infection. Next is the serological test. Serological tests such as enzyme-linked immunoabsorbent assay or as known as ELISA may be used to detect specific antibodies against M. Yokogawai in serum. These tests can be helpful in cases where stool examination is inclusive or unavailable. The treatment of Metagonimus Yokogawai infection typically involves the use of anthelmintic medication, with praziquantel being the drug of choice. Praziquantel is highly effective against a wide range of trematode infections including Metagonimus Yokogawai. Prevention and control of Metagonimus Yokogawai infection involves several strategies aimed at reducing the transmission of the parasite and minimizing the risk of human exposure. So here are key measures for prevention and control. First is health education and awareness. Raising awareness among communities and individuals involved in fishing and food preparation about the risks associated with consuming raw or undercooked freshwater fish. Next is safe food handling practices. Ensure thorough cooking of freshwater fish to kill Metagonimus Yokogawai larvae. Fish should be cooked to an internal temperature of at least 145 degrees Fahrenheit or 63 degrees Celsius. Discourage the consumption of raw or undercooked freshwater fish, especially in endemic areas. Next is sanitation and hygiene. Hand hygiene is really important, with soap and water before handling food, after using the toilet, and after contact with potentially contaminated water sources.